Good morning, everyone. 11:05. We're going to go ahead and get started. Welcome, everyone, to Grand Rounds. My name is Rebecca Muley, and I'm one of the Grand Rounds co-directors, along with Doctors Adi Talati and Alexander Harris. Next week, our speaker is Dr. William A. Carlazon Jr., professor of psychiatry and neuroscience at Harvard Medical School and the Jerry and Phyllis and Rappaport Chair in Psychiatry at McLean Hospital. The title of his talk is Preclinical Studies of Stress and Depression, Enhancing Translational Relevance. Before we introduce today's speaker, a reminder that if you are participating via Zoom, please post questions using the Q&A feature, not the chat feature. As the online moderator, Dr. Talati will forward questions to me for our speaker to address. We no longer have our online participants contributing via telecamera, via Zoom, but we do address all of them that we can in the auditorium. We encourage trainees to consider asking a question, and we will hold a space for trainee questions in person and online to go first. Please also remember to fill out your CMA surveys after the talk if you would like CME, CE, or CS credit for today's lecture. There's a QR code in the table outside for those in person. The link is posted on the chat on the Zoom at 12:15 p.m. Finally, we would like to thank Dr. Robert or Bob Remian for his generous personal donation that allows us to have our morning pastries and coffee, as well as the trainee luncheon following our grand rounds. Today's grand rounds will be given by Dr. Brian Dias, the Associate Professor at the USC Keck School of Medicine and the Children's Hospital of Los Angeles. I would now like to ask Dr. William Pfeiffer, Professor of Medical Psychology and Psychiatry and Pediatrics at CUMC and the Chief of the Institute for Developmental Sciences, to introduce our guest speaker.
I'm glad so many of you made it today. We were a little afraid with all the commitments that have to be made with students being on break, et cetera. But so thank you, and thank you wherever you are in Zoom for being able to join us as well. So Brian Dias comes to us from the Department of Pediatrics at the USC Keck School of Medicine, and he also directs his research laboratory at Children's Hospital of Los Angeles. He and his team study the legacies of stress and how they echo across generations. He's very committed to educating and mentoring the next generation of scientists. He's been awarded multiple grants for mentoring students at all ages, including high school students, students at the USC scholars enrolled in neuroscience graduate program in the medical school. I really found particularly interesting, and I think you probably still do this, your longstanding teaching of Tibetan Buddhist monastics. Really, I'd like to hear more about that. He's intimately involved with developing education curriculum, particularly for Minnesota-based family-wise services. They really aim to strengthen families by promoting the well-being of children. A very successful operation. Just very brief training background. Earliest training was in Mumbai in molecular biology, neurosciences at the University of Texas at Austin, then had his first Howard Hughes Medical Institute postdoctoral fellowship in neurogenetics with Bruce Baker, then had a second one in psychiatry with Carrie Ressler. His grants are numerous and impressive, including from NIH, 2 current grants, as well as a grant from CFAR. I could go on, as is my wont, but I won't. But one final accomplishment I'd like to highlight is his Howard Hughes Medical Institute fellowship with his graduate student on a program to ensure that students from historically excluded and underrepresented in science are prepared to assume leadership roles in science and science education. A tough time to be doing that, but Brian continues that fight. And so we thank you for that. And thank you for coming all the way to join us today.
Hi, everyone. Good morning. Thanks so much for choosing to spend your time listening to this talk in person this morning. And thanks to everyone online for joining as well. I know we're living in extraordinary times. I want to thank Bill for the invitation to visit, especially at this time. And hopefully we can talk about some science that resonates with all of us, and also end with the silver linings and about how we can end legacies of stress and trauma. By way of disclosures, I have nothing to disclose, and I just want to remind everyone about my learning objectives. I hope you walk away from this talk appreciating the roots via which legacies of stress echo across generations, are able to discuss the cellular and molecular vehicles that relay the batons of stress from one generation to the next, and then also appreciate that the die is not cast, that there's a malleability in our biological systems that can be leveraged to halt legacies of stress from perpetuating across generations, but also allowing for us to engineer legacies of flourishing. I do want to say before I start that there is disturbing imagery and dialogue as part of this talk, and should that upset you in any way, please feel free to take care of yourself as you think best. They mess you up, your mom and dad. They may not mean to, but they do. They fill you with the faults they had and they add some extra just for you. But they were messed up in their turn by fools in old-style hats and coats who half the time were soppy stern and half at one another's throats. Human, hands-on misery to human, it deepens like a coastal shelf. Get out as early as you can and don't have any kids yourself. These words by Philip Larkin eloquently give voice to the idea that stress affects not only the individuals who are directly exposed to that stressor, but also future generations. Legacies of stress echo within and across generations, and my lab is interested in understanding what do these legacies look like, how do they manifest, and how do we break them? So in thinking about what legacies of stress look like, these are shocking statistics from the California Child Welfare Indicators Project, which shows the incidence of different levels of abuse that have been reported to the child welfare agencies. And as you can see here, in these alarming statistics, there's a high prevalence of general neglect and physical abuse. And of course, we know through the landmark ACEs study that there are lots of adverse childhood exposures, and there's a variation in how many childhood exposures individuals experience, with, in that particular study, 12.5 individuals, percentage of those individuals experiencing 4 or more adverse childhood exposures. And those childhood exposures are placed into buckets, which are abuse, household challenges, and neglect. And the long-term ramifications of these childhood exposures, of course, is well known, where there's a compounding effect where you have increased exposure to childhood exposures, increasing the potential incidence of disorders like diabetes, like respiratory disease, as well as stroke. Now, if all of this was not grave enough as is, we're now appreciating from historical events and atrocities, data collected in the aftermath of those events and contemporary biology, that legacies of stress don't only affect that generation directly exposed to them, but also future generations. So take, for example, Rachel Yehuda's work showing that babies who were in utero at the time that their mothers directly experienced the 9/11 terrorist attacks and went on to develop PTSD, those babies had lower cortisol levels at age 1, lower stress hormone levels at age 1, compared to a comparable cohort of babies whose mothers experienced the 9/11 terrorist attacks but did not develop PTSD. Why this is important is because Rachel's group has also shown that descendants of Holocaust survivors have, even though they did not experience the Holocaust themselves, have lower levels of cortisol, which increases the incidence of PTSD and anxiety. Then you have work from Tanya Jovanovic and colleagues who've shown that babies born to mothers who were abused, even if that abuse happened preconceptually, those children now brought into the laboratory present with higher anxiety and depression-like metrics. And then, of course, we know through famines that have occurred across history, both natural as well as anthropogenic, like the Dutch hunger winter in World War II, like the Överkalix famine in Sweden, and other famines, that metabolic access to food affects metabolism not only in the generation that's directly exposed, but also future generations. Again, all of these giving voice to this idea that legacies of stress echo within and across generations. So how does this happen? We now know that there are really three routes via which negative environmental experiences have ripple effects across generations. They affect the generation that's directly exposed and then ripple out into the G1, G2, and several other generations. You have the germline, which could be altered. You have in utero development that could be altered if the stress or trauma is experienced during pregnancy. And you could have altered care being bestowed upon the next generation that could be perturbed because of a stressful event. And now we also know that there are a myriad of biological vehicles that allow for these ripple effects to perpetuate across generations. And I'll touch upon a few of these during my talk. So you have, through the germline, you have genetics and epigenetics, which is one vehicle by which germline legacies of stress perpetuate across generations, extracellular vesicles. You have placental biology, brain development, and immune function, which is affected during in utero development by stressors. And you have the cortisol system, the HPA axis, that is a key axis that is affected by altered care being bestowed upon generations as a consequence of negative environmental experiences. So what I'm going to do today, this morning, is talk to you about three stories. In the first story, and most of my talk is going to focus on germline legacies of stress using our work in mice and using our work in olfaction to study germline legacies of stress. Next, I'm going to focus on fetal hyperglycemia in the mouse and talk about how that would affect future learning and memory. And then I'm going to close out this talk by talking about some of our work in rhesus macaques, where we leverage naturally occurring infant maltreatment, so altered care being bestowed upon rhesus macaques, to talk to you about some enduring legacies of that maltreatment, of early life adversity, and how it comes to become embedded under our skin. Now, I list all of these as though they're independent entities of each other. That's partly because we're studying them independently, but I don't want you to walk away from this talk without knowing that really they're intricately connected to each other. There's a relay that's happening. And so you could envision a scenario wherein a preconceptual stressor affects in utero development and how the embryo develops. Or you could have altered care being bestowed upon an individual, affecting how that individual's germline responds to that care. And so we don't want to be viewing these as independent, although the stories that I'm going to be talking to you about are independent of each other. Of course, I stand before you just as an ambassador. I still do experiments in the laboratory, but most of the work is done by other individuals in the laboratory. So the work that I'm going to be talking to you about on germline legacies was led by Hajawed, Soma Sanigrai, and Sarah Hunter while at Emory, and then picked up by Nabil Bhindarwala, Dr. Rashmi Joshi, Dr. Nancy Wu, and Kathy Liu, who are studying these germline legacies. The story about fetal hyperglycemia and high fat, high sugar in the mouse is led by Samantha Stack, who's a graduate student in the lab. And then the story about infant maltreatment in the rhesus macaques is led by Laura Korobkova and is a collaboration with Dr. Mar Sanchez at the Emory National Primate Research Center. But there are also many other individuals in the lab whose work I would not be able to talk to you about today, and they're on this slide. Happy to talk about some of their work, which ranges from glucocorticoid receptors to learning and memory to Prader-Willi syndrome as well. So let's get into it then and talk about germline legacies of stress. So in asking this question, how does stress echo across generations? To me, it always seemed like we were looking for a needle in a haystack. And our job was to try and shrink that haystack and make that needle much larger. And I posited many years ago that maybe we could leverage the power of smell to shrink that haystack and make that needle much larger. And the reason we can do that, of course, is no surprise to you, because the seminal work, the foundational work, was done here by Drs. Richard Axel and Linda Buck, who showed us that there's an exquisite structural and functional specificity of the olfactory system. I know this is known to most of you in the audience, but in case not, I'm just going to go briefly over the olfactory system. Within the nose, you have olfactory sensory neurons that reside on the olfactory epithelium. These sensory neurons send their axons into the brain, and they coalesce into functional units called glomeruli in the olfactory bulb. Now, there are about 1,200 different genes that encode for odorant receptors, but a single olfactory sensory neuron only expresses one of those 1,200 odorant receptors. So of all the colors of the rainbow that this particular olfactory sensory neuron could express, it expresses the red odorant receptor. Of all the colors of the rainbow that this particular olfactory sensory neuron could express, it expresses the blue odorant receptor. Why is that important? That's important because no matter where in the olfactory epithelium, the olfactory sensory neurons that express the red receptor reside, they all send their axons into the brain, coalescing into the functional unit called a glomerulus. And why is this important? This is important because in the laboratory, we have transgenic mice that allow for us to stain specific olfactory sensory neuron populations that express that particular odorant receptor. So shown here is the M71 LAC-Z mouse, where we have a mouse where all the receptors expressing OLFR 151 or M71 receptor can be stained as blue. So you can see the puncta here where you have blue sensory neurons in the main olfactory epithelium, sending their axons into the brain, coalescing into the glomerulus in the olfactory bulb. Complementing the structural visualization of the olfactory system, we know that acetophenone, an odor which has been called cherry blossom in the press, it smells like almond to me, that activates the M71 olfactory sensory neurons, while another odorant, propanol, does not. And what's important for me to emphasize to you is we also have other mice in the laboratory, like the MOR23 GFP mice, where the neurons that express the MOR23 receptors are stained green, and the odor that activates that particular odorant receptor population is called lyral. So I posited way back when that we could potentially tag stress using olfaction. And then because the olfactory system has this functional and structural specificity that I talked to you about, maybe, just maybe, the germline, the genes that encode these odorant receptors in the germline would bear influences of that tagging of stress in a G0 generation. And then maybe, just maybe, olfactory neuroanatomy and behavior would be affected in that next generation. So we want to tag a stressor in one generation and follow it across generations, shrinking that haystack, making that needle much larger. And so what we've shown now over 3 papers is we've shown that, shown on the y-axis is something called odor potentiated startle, which is a measure of sensitivity to the particular odor. And what we've shown is that the F1 animals who are sired by F0 males, whose fathers were just hanging out in their home cage, those animals show X amount of startle to acetophenone. But animals whose fathers had been conditioned to acetophenone show a higher sensitivity to acetophenone. And animals whose fathers had been conditioned to propanol show a higher sensitivity to propanol. Now, contrary to what one might have read in the popular press, we do not call this a fear of that particular odor. Mice do not inherit the fears of their fathers. This will be important as I go through my talk. I'm firmly in the behavioral sensitivity space, right? Complementing this behavioral sensitivity, we want to look at what the neuroanatomy looks like. And so shown here are whole mounts of M71 LAC-Z neurons and the glomeruli in the olfactory bulb. This is what the glomeruli look like in M71 LAC-Z animals that have been sired by F0s just hanging out in their home cage. This is what the M71 glomeruli looks like in F1s whose fathers were conditioned, F0s were conditioned to propanol. Remember, propanol does not activate the M71 receptor. In odor-naive animals who've never experienced acetophenone before, but who've been sired by F0s who've been conditioned with acetophenone, you have an enhanced representation for M71 glomeruli and M71 neurons in the brain. And we know this to be the case because we know that there are more neurons that express the M71 odorant receptor in these particular animals. And I do want to point out the beautiful work led by Clara Cliff, who's in the audience, from Dr. Bianca Jones-Marlin's lab, who's also in the audience, who've shown with beautiful iDisco work and an enhanced neuroanatomical representation of a particular odorant receptor in the F1 generation after F0 conditioning with that odor. So what we were able to do then is we were able to tag a stress in one generation and then follow it into the next generation, showing that there's a behavioral sensitivity in the next generation to that odor and an enhanced neuroanatomical representation. I need to take a step back to also talk to you about work that was done in Dr. Kerry Ressler's lab by Seth Jones, where in 2008, they put out a paper showing that in F0s, when you condition the fathers with acetophenone, and just look at the fathers, they have an enhanced representation for that odorant receptor in their own brains in the F0 generation. And once again, this was beautifully demonstrated by Clara and Dr. Marlin in their paper. So we're at a point now where we have like father, like son. So the question is, how is that happening? So we typically think this is happening in two ways. There could be a social transmission of information where dad is telling the fetus through mom how to respond in a certain way. Or we could have an inheritance, a biological inheritance of information that occurs via the germline. We did a lot of experiments over the years to tease this apart. And what emphasizes to us, in addition to number 2 and 3, which are looking at the F2 generation and cross-fostering, that this is an inheritance, not a social transmission phenotype, is that we can perform in vitro fertilization where there's no traditional mating, and we still recapitulate all of our effects. So the question now becomes, like father, like son, but how? And so we looked at DNA methylation, et cetera, in the sperm, and were able to show that there were DNA methylation changes in the sperm of the animals who had been conditioned to acetophenone at the M71 specific locus. But in that paper, we also suggested that this provides a rationale for the sperm registering information about that experience, but not a mechanism for how this is going through. And so that moves me to, how is this actually happening? And the reason we posited that it really couldn't be DNA methylation that allows for information for this baton to be passed from the one generation to the next is because for the most part of epigenetic reprogramming, methylation marks are scrubbed clean at 2 different stages of development as the embryo develops. Now, we now know over all these years that there are certain genetic loci that escape epigenetic reprogramming. 25% of loci in primordial germ cells do escape epigenetic reprogramming. Most of those are associated with obesity and schizophrenia. We don't know the logic about why something escapes reprogramming versus not. So DNA methylation could well be one mechanism by which information is going from one generation to the next. But we posited way back when that RNA in sperm can be found as conductors of gene expression that allow for this baton of stress to be handed from one generation to the next. And these are emails you save to show your students and everyone else. One of the reviewers basically said that is not the way biology works. Sometimes we don't listen to reviewers. Most of the time we do. But what we now do is we routinely inject RNA from mice who've been conditioned to a particular odor, say odor A, into naive single-cell zygotes, put those zygotes back into a surrogate female, get the offspring. And what we do is we recapitulate all of our effects just by putting the RNA from stressed sperm into naive zygotes, we recapitulate the sensitivity to the odor and the enhancement in olfactory neuroanatomy. I want you to just note when we published this paper, 2020. Beautiful work by Tracy Bale, Ollie Rando, Chi Chen, Isabel Mansui, and several others prior to 2020 have shown in their models that RNA are bona fide carriers of information from in sperm, are bona fide carriers of preconceptional information from one generation to the next, using the protocols that I just talked about. What they did is similar, either in the nutritional space or the stress space. They inject RNA from sperm of mice, male mice, who've been exposed to those particular stressors, and then recapitulate all of their effects as if it was a traditional mating. So then the question becomes consequence. What consequence does this have for the next generation? And this is where that learning objective about there's a malleability to the system, the die is not cast, there are silver linings comes through. Because what we posited is that, remember, I call it a behavioral sensitivity, not a fear. That was intentional, because the behavioral sensitivity, we posited, primes the system to now respond to future encounters with that, in this case, particular odor. So what we do is we took these F1 animals, and now who've either been sired by F0s who've just been exposed to an odor or who've been trained with fear conditioned with an odor. And then we take those F1 animals and we give them only a single odor shock pairing in a particular context. So that's a sub-threshold learning paradigm. And then we test whether that animal remembers that single odor shock pairing in terms of memory consolidation the next day by examining its freezing response or odor potentiated startle response to that particular odor the next day. And what we find in female F1 offspring whose fathers had been conditioned to acetophenone, they show a consolidation of memory, that single odor trial fear conditioning, as do F1 lyral females whose fathers were conditioned to lyral. We don't see that in the males. We don't know why that is, but this suggests to us that there's a positive, there's a silver lining for the next generation. It's not all gloom and doom. It primes the system to respond appropriately when they encounter certain stimuli in their own environments. So to summarize this part then, I've shown you that there are intergenerational influences of stress that have been demonstrated in the human as well as rodent conditions, and they can be studied by leveraging the power of olfaction. These are mediated in part by RNA in sperm, and I say in part because there are other mechanisms probably out there which we don't know about, and they confer adaptive properties to the offspring, that it's not all gloom and doom, there are positives as well. And if you'd like to read a little bit more about how chemosensation has been used to study legacies of stress, this is a review that I wrote last year, which talks about multiple species being used in this space to be able to really tag stressors or tag exposures in one generation and follow them across generations. So that was story number one, germline legacies in mice. I'm now going to turn my attention to altered in utero development and not talk about what's happening in utero, but where the exposure is in utero. And we're going to do that through the lens of food consumption and the quality of diet. So this is what the USDA dietary recommendations are in terms of what one should eat. And as you can see, what the actual consumption of food is completely different. So much so that this food pyramid, which should actually be inverted, looks like this, where most of our calories come from highly processed food, high in fat and high in sugar. Now, most studies that have looked at fetal hyperglycemia and the consequence of high fat, high sugar diet, which might contribute to fetal hyperglycemia, has looked at type 2 diabetes or the development of diabetes and obesity in offspring. But we know that there are altered trajectories of development, trajectories of learning, in the next generation as well. And we wanted to see whether we can model this in the laboratory and put some neurobiological texture to it. So how do we do this? We leverage naturally occurring developmental trajectories of learning in mice. So just like humans, which exhibit a naturally occurring developmental trajectory of learning, mice do too. In this beautiful paper by Adam Ramsaran from Paul Franklin's group, what they showed is that there are 20-day-old mice, P20, versus P24 mice. They take these mice and they train them in a particular context, context A, where they pair that context with 3 unsignaled foot shocks. They then bring those animals in 1 day later and present them with either context A or context B. P20 animals show a generalization of memory to both context A and context B. They freeze more to both those contexts. Versus P24 animals, which show a specificity of response, where they respond and show high freezing only to A and not to B. So there's a transition from generalized to precise memory. If we wanted to figure out whether there were developmental trajectories of learning that were different based on high-fat, high-sugar diet, we thought we needed to shrink that developmental window a little bit because P20 to P24 is a long time. And so what Sam did in the lab is she tried to figure out what are the boundary conditions, where does this transition happen? And what Sam focused on was the P21 to P23 animals. And what she found is using a similar strategy, exactly the same that Adam and Paul did, which is train animals in context A, then bring them in and test them in context A and B. You can see here what Sam is showing you is that P21 animals show a generalized memory because they freeze the same amount in A and B, versus P23 animals show a specificity of response by freezing only in A. So now that we have this developmental trajectory of learning from generalized to precise, the question now becomes, can we induce fetal hyperglycemia? One way by which we can do that is by administering a high-fat, high-sugar diet during gestation, and then look at the next generation with the P21 and P23 animals. A key part of in utero work or legacies of stress work is that it accelerates brain development or it decelerates brain development depending on the time of exposure. There's been a lot of work done on the accelerating, that is, windows of opportunity closing too quickly. But there's enough evidence to suggest that if the exposure happens in utero, there's actually a slowing down of maturation. And that's what we seem to maybe see here. So I'm going to walk you through these data. Here again, we're doing high-fat, high-sugar during gestation. The pups are born. They're put on a controlled diet, and then we're testing them at P21 and P23. So as shown here is a measure of generalized to precise memory. The higher the bar, it's more precise memory. And as I showed you earlier, P21 animals show more generalized than precise, and P23 animals inch their way up to precision. But what we find is that P23 animals who had experienced that high-fat, high-sugar diet while in utero, they look like P21 animals in showing generalized memory. Our interpretation of this unpublished and very preliminary work is that that exposure to high-fat, high-sugar diet is slowing down the cadence of development such that a P23 animal is now looking like a P21 animal, and therefore there might be neurobiological substrates which appear like a P21 animal, therefore supporting generalization and not precision of memory. So how do we start doing this? We use these mice in the laboratory and this technique called trapping. We have force trap mice in the lab where if we administer them tamoxifen before training, we can stain cells that are active at the time of learning, of encoding, as red. We can then bring them in and test them the next day in either of these contexts and do CFOS staining to figure out what cells are active at the time of retrieval. And so shown here is a mouse brain where you have the counter stain, which is hooks right here to tell us where we are in the brain. You have all of these cells that are labeled red, which are the cells that are active at the time of learning, of encoding information. CFOS, which is in green, gives us what brain regions and what cells are involved in retrieving that information. And yellow gives us what cells are involved in both. Those engram cells that are active, ensembles that are active at the time of encoding and are reactivated at the time of recall of information. We're doing some analyses right now to say, are engram profiles changing in animals that have had this high fat, high sugar exposure during gestation to allow for these developmental trajectories of learning to be skewed, to be slowed down because of this particular experience in utero. So that was the second story with fetal hyperglycemia in mice. The last story that I want to talk to you about is this idea that one of the vehicles that allows for stress to get under the skin is early life adversity and altered care. I think we all appreciate, through the wonderful work that has been done by colleagues in the rodent space as well as in the human space, that parental behavior is a huge component. This is, again, where Dr. Bianca Marlin's expertise comes in. Beautiful work showing parental behavior really can sculpt future biology and psychopathology. So I'm going to leverage our work and a collaboration with Dr. Mara Sanchez at the Emory National Primate Research Center to talk about infant maltreatment in rhesus macaques to answer the question, are there enduring novel signatures of early life adversity that might hold promise as biomarkers, as well as point to the etiology of the adverse effects of early life adversity? So what we decided to do for this is we decided to say that one criteria for the specimen that we wanted to get was we wanted to be agnostic to where and what tissue types, et cetera, were being affected by early life adversity. And so we landed on extracellular vesicles. Extracellular vesicles, or EVs, are these tiny little blobs circulating through our blood. They're released from almost all organs and tissues. They're being taken up by almost all organs and tissues. And we thought that extracellular vesicles could give us a readout as beacons of what's happening organismal-wide. And interestingly, we focus on extracellular vesicles also because extracellular vesicles are one mechanism that has been posited to allow for information about the environment to get into sperm. So there are these little extracellular vesicles specifically in the epididymis. Epididymis is the place where the sperm hang out before they're ejaculated. There are all these epididymosomes. Try saying that 20 times. These epididymosomes that are chock full of small non-coding RNA. Those non-coding RNA get shoved into these sperm. They then fertilize eggs, and that's how you get information, that RNA information passed from one generation to the next. And that's, again, beautiful work from Ollie Rando, Chi Chen, as well as Tracy Bale. So we posited that extracellular vesicles could be one mechanism by which information is traveling through the highways in our body, that is the circulatory system, to affect the whole organism in the shadow of early life maltreatment. So to do this, we leverage naturally occurring infant maltreatment in the rhesus macaque. So I want to emphasize that again. This is naturally occurring. So 2 to 5% of monkey mums will maltreat their young. That maltreatment consists of physical abuse and high infant rejection. Let's go back to that California Child Welfare Indicator Project. And those were the buckets of adversity that were highly predominant there. It involves poor maternal care early in life that is extremely stressful. And our question was, can we get blood from these animals who've been maltreated as infants during the first 3 to 6 months of life? Can we get that blood in adolescence? And look at extracellular vesicles and see what those have? And could those tell us that something that happened in the 3 to 6 month period still lasts at the adolescent time frame? So what we do is we get these extracellular vesicles with the help of the CHLA extracellular vesicle core. We use density gradient centrifugation and size exclusion to get really pure extracellular vesicles. And then we perform RNA-seq of the contents in these vesicles. And what we find is that RNA related to immune function and mitochondrial energetics are the ones that pop up for us that are either highly expressed or suppressed in individuals who've been maltreated as infants versus not. And I was having a conversation this morning about mitochondria, and I think that's the next frontier with respect to, we're already tapping on its doors with the amazing work by Martin Picard and colleagues, where you have mitochondria as being this organelle that is affected by a whole slew of stressors, including, for example, early life adversity and maltreatment. So this was great. I want to acknowledge the fact that these are 7 animals per group, so we're very underpowered, so we're not making terribly much of this. But what was surprising to us, what we were very excited about, what is probably going to be very controversial, is my next slide. Only 30 to 40% of our reads align to the macaque genome. So what Laura did is she threw the reads that didn't align to the macaque genome onto the microbiome genome. And there's a ton of microbiome-associated RNA in our extracellular vesicles. So rightly so, the reviewers want to know, is this real? And we're doing some experiments to determine that, because we want to dot our I's, cross our T's. I can only handle one controversy in my career. But what we are finding, and again, I want to emphasize that this is a small sample size, but there are changes in the microbiome, in the RNA associated with specific species, depending on whether an animal was not maltreated versus an animal that was maltreated. So what we're trying to find out now is, do extracellular vesicles actually contain microbiome-associated RNA? And if they do, then for us, the next questions are going to be, what is that RNA doing when it gets to its recipient cells? But I think in general, for this field of inquiry, if EVs are this next molecular frontier that we are seeing as a way through which biological adversity gets, or so negative environmental experiences get embedded under our skin, we still have to grapple and wrestle with this problem of where are extracellular vesicles coming from and who's taking them up. And right now, it's a free-for-all. So we need our technologies to now start trying to keep up with being able to tag extracellular vesicles, maybe being able to follow them as they go from one place to the other. So that's a lot of microscopy. We are taking a different approach, collaborating with Dr. Stacy Bilbo, who's interested in the lung. And what we are doing with Stacy's help and collaboration is we're looking at what happens, what the extracellular vesicles are when specific cells in the lung are ablated. The idea being, if that's the focal point of origin, maybe extracellular vesicles will tell us that they were coming from the lung. From a differential perspective, we can then make some guesses as to what was coming from the lung and how that's affecting the brain using some approaches that we're leveraging in the mouse. So with that said, I'm going to briefly go back to this slide with my agenda saying I've talked to you about 3 routes by which germline legacies of stress echo across generations, how an in utero stressor like high fat, high sugar affects developmental trajectories of learning in the next generation, and how infant maltreatment in the rhesus macaque and EVs that we've got from these macaques give us some idea into the sinister effects of early life adversity. But again, I want to emphasize that there's a malleability to this system. It's not all gloom and doom. So legacies of flourishing can be engineered. And the one thing we know from the literature that allows for us to halt legacies of stress and engineer legacies of flourishing is unfortunately something that is a scarce commodity these days, and that is stability. And that stability is economic stability, That stability is environmental stability through various axes, and that stability is through caregiver and social stability as well. So as I said, I stand before you as an ambassador for these amazing people who've done the work. There's many more shoulders that I stand on. And I always end my talk with this slide, and I apologize in advance. It's incomplete. Some people come up to me at the end of this and say, my name wasn't on there. It's an ever-growing list. I apologize. But it's just amazing to be here at Columbia because I can't tell you, there's so much shade thrown on other people's work. I am informed and inspired by the people on this slide and many more. Their work informs and inspires our work. I stand on their shoulders. Many of them are in this audience or at Columbia. I thank them for informing and inspiring us. I thank you for your attention, and I'm happy to take any questions.